to, in a eukaryotic cell, once the genetic information is in the host cell, it will depend upon what type of genetic information as to where the events are going to occur. They may occur in the nucleus, or they may occur in the cytoplasm, depending upon whether it's DNA, which would go to the nucleus, or RNA, which would go stay in the cytoplasm. And of course, right, because we are talking about, a mechanism related, though viruses aren't living, right? We do see those odd situations where things such as HIV, which is an RNA virus, so you would think, oh, everything's happening in the cytoplasm, but in this particular case, that RNA is reverse transcribed. Once it gets into DNA, the DNA goes into the nucleus. Okay, so everything happens in the nucleus for that particular organism. For that particular virus in that organism. All right, and then we also have the envelope. The envelope comes into play with animal viruses. Bacteriophages don't have envelopes. Okay, so the envelope is going to come into play as something where they're different, because in order to obtain that envelope, it is getting it from those phospholipids that were originally part of the cell membrane. Okay, so that requires a budding process. Budding is a process that can't occur if you have a rigid cell wall, right, which prokaryotic cells have. So we're not going to see that particular stage of the viral replication for bacteriophages. One, because they don't have the envelope. Number two is there's no way that they could have an envelope. There would be no way for those those bacterial viruses to exit the bacterial cell that has a cell wall. And then the other is the cell wall plays another aspect in the sense that the bacterial cell wall prevents the nucleocapsid from entering the host cell. So in the case of a bacteriophage, a virus that infects bacterial cells, the capsid, that protein encasement, right, that protects the genetic information while that virus is outside of the host, is going to stay outside of the host. Only the genetic information is going to enter into that cell, okay? Versus in an animal virus, it is possible that the entire nucleocapsid is going to be, through endocytosis, is going to be brought into the cell, and then uncoding happens within the cell. That's a possibility when it comes to animal viruses. That's another difference that we can see between the two. So those the differences are, based upon really the differences that we see in regards to, primarily in regards to the host cell, but occasionally due to what the virus actually has, okay, as part of its structure. Then we'll just do a little talk about the role of viruses in the cancer development. So viruses have been recognized to play a role with cancer. There are 5 viruses, the hepatitis B, hepatitis C, the human papillomavirus, Epstein-Barr virus, and the human T lymphotropic virus, type 1, okay, and this is in that second item that was to be read, that are going to contribute, are the key ones that contribute to cancers. Most of those are DNA viruses, okay, with the exception of hepatitis C, which is an RNA virus, but it has a DNA intermediary. Okay, so in the case of these viruses, their role in cancer development, it can vary. It can either be a direct role in that process or an indirect role in that process. Okay, but we do find that as far as cancers are concerned, cervical cancer and liver cancer, 80% of those are actually caused by viruses. Okay, for liver cancer, you're looking at hepatitis B and hepatitis C being the main causes of that. And for cervical cancer, then we have the human papillomavirus. Now, the role that they play is as follows. DNA, for your viruses, some DNA have the ability to, DNA viruses have the ability to incorporate into the genome, okay, much like we saw with the prophages, right? In the case of an animal virus, when the DNA of the virus inserts into the genome of the host, that is referred to as a provirus. The provirus insertion is one in which is a permanent thing. Once it's inserted, it does not have the ability to excise out of that host genome. Viruses have promoters in front of their genes that are very strong, that are going to promote replication of that virus to get those capsomeres produced and any other specialized proteins that are necessary to assemble and crank out a lot of viruses by that host cell. So when the DNA inserts into the genome, it's the where it inserts sometimes can place that promoter in such a position that it now is going to result in the transcription of genes that shouldn't be transcribed. So now all of a sudden we can see what we call the lack of cell cycle control. Some of these very important proteins that are involved in cell cycle control now are not going to be, either they're not made properly or too many are being produced at a time when they shouldn't be, so it throws cell cycle control out of whack, okay? Another aspect that can happen is a long-term damage. The virus in itself, so we see with, say, hepatitis C, that's a long-term, so a chronic infection, is one where the damage occurs over an extended period of time, and of course, liver cells can replenish, right? They can replace, right? You can take a portion of your liver, and those cells will reproduce again in order to replace the lost tissue. And in that process, though, while that damage is occurring and replacement is happening, so those cells, the host cells are replicating, there's a higher chance of just spontaneous mutations, right? Spontaneous mutations being mutations that occur naturally, that aren't caught. And so during that repair process, those type of mutations can, there's a higher probability of those type of mutations being introduced and therefore causing those cancerous cells. Okay, those specific genes referred to the oncogenes that are more susceptible to these sorts of changes that result then in cancer. Another way in which they can affect would be kind of another indirect way, which would be to suppress the immune system. Okay, the immune system, part of our immune system, we have cells that are involved in this process that are going to be monitoring not just for foreign material that comes into the body, but they also monitor for cells, our own cells, that are displaying indicators that something's wrong with them, right? And cancer cells, because of their quick division, do display markers indicating that something is off. And so when the immune system is repressed, then those cells aren't going to be, we're not getting the response that we need from those cells, so that our immune system is actually being able to identify the cancer cells and destroy them, right? And if our immune system's not doing that, that leaves them unchecked, okay? So those are different ways in which viruses can play a role in cancer development, and we do recognize that they are a cause of cancer. They're not the only cause, right? There are various other reasons why an individual will get cancer, but viruses play a role in some of the cancers that are out there. And then the last section for this particular unit is we're just going to talk about is the prions. The prions are acellular because they are not a cell, right? They are actually a protein, literally a protein. They are referred to as proteinous infectious agents because it is a protein. Prions are actually naturally found within most mammals. When folded properly, it has its function and plays a role in the body. That 3D structure folding of that protein, it is actually on the secondary level where we see when it's properly folded, it'll have many of these alpha helixes. The infectious form of this particular protein is one that is improperly folded. And when it's improperly folded, that folding in the secondary structure tends to have more beta pleated sheets instead of alpha helixes. And so when you have this improperly folded prion, which in this case they're identifying as the PRPSC here. That's the improperly formed. What happens is because proteins cannot reproduce, right? There's no such thing as reproducing of proteins. But what these proteins do, these improperly proteins do, is that they interact with properly folded protein, causing them to refold in the improper manner, okay? And therefore, the numbers of the improperly folded prions increase. And they will start to aggregate, these improperly folded proteins will aggregate in the brain. And thus where we get the disease, okay, you get these large vacuoles forming in the brain, and if you take a brain biopsy, what you're going to see is a spongy-like appearance, which is what you see down here, in that brain tissue, okay? The protein can be obtained through ingestion of contaminated material that has the improperly folded prion, through transplantation, okay, or contact with mucous membranes of an individual who has this condition, or it can be a genetic, okay? So the diseases that are out there for the prion, if you're talking about human, then for human diseases, you'll hear it as the Creutzfeldt-Jacobs disease. That's the more commonly recognized in the US. The Gertzmann-Schlauser-Schlenker syndrome, which is what you tend to hear more in Europe. They also have fatal familial insomnia as well as Kuru. Now, Kuru is not a commonly recognized anymore because it is with a certain group of indigenous individuals who practice cannibalism as part of their ceremony for people who die. And so once we discuss, with the understanding of prions, and we educated in order to stop that practice, right? That kind of eliminated in that, but it was limited to a group of a very specific population. Now, it is also found in many other animals, okay, because again, most, mammals have this protein, okay? So it was actually first identified in sheep, it was called scrapie, which is why you'll notice that the symbol, they said the PRPSC was for scrapies, because that was where it was first identified. But since then, of course, it has been identified in the mink population, so it's transmissible mink encephalopathy. For them, chronic wasting disease for deer, and so it is found in the U.S. In the deer population. So if you're a deer hunter, right, there's certain tissue you do not consume to be on the safe side. And then you've got the bovine spongiform encephalopathy, which got a lot of recognition for what we refer to as the mad cow disease. When they first saw that in the population of the bovine, like in a herd, they would not use the meat for human consumption, but they proceeded then, though, because it's a money source, it's a loss of money, so they would put it into other food sources for other animals. That has since, again, once education happened, we recognized what was being transmitted, more information about it. Now, should they see even a single animal in a herd that's displaying it, they will kill the herd and they burn, they incinerate. Because this particular protein is very hard to get rid of. It does not denature very well. So unlike many other proteins where denaturation is it can be as simple as high heat, this one is requires the use of the autoclave, so very high heat, but not just autoclave. It needs the material needs to be put in concentrated sodium hydroxide and in the autoclave. So high heat and high base in order to deactivate it. There is now an enzyme called Priazyme that has been, I don't remember if it was somehow produced or where they found it or identified it, but it does have the ability to remove. It is one that is used, an enzyme that can be added to be used to break down that improperly formed prion. Okay, so that's a newer product that has been used. But other than that, there is no cure for this, right? And because of the fact that it's so difficult to get rid of, they usually go by symptoms, which would be standard for individuals who are having a infection of the brain. Okay, and then they would and then they wait until after death to actually confirm, because they don't want this to get out in the surgical rooms, just because the difficulty in actually disinfecting the room from this particular protein, and so they'll wait until, and of course in a morgue, it doesn't matter, right? Because everyone going in there is already dead, so they're not worried about transmission. Okay, so usually biopsies are done after the fact. Again, there is no cure. There's nothing that they can do for this particular disease, at least not at this point in time. All right, so that covers my slides for this. Yes, it is. That covers my slides for this one. And so what I've got for you guys is a game. I'm required to have an activity. So I figure in-class activities are best we play. And so I've pulled together questions that I feel you should be able to answer having gone through this unit. That's why you'll notice all these in-class activities are the day before, or the lecture before the exam. What is today, the 17th, right?
Yeah.
I divide the class in half, but you guys are not playing to my favor here because that side looks like it has fewer people than this side. So I have 2, 4, 6, 8, 10, 2, 4, 6, 7. So let's see if I can get at least one person on that side to even it out just a tad. We have an odd number here, which is fine. It actually makes it better so you're not always against the same person. And what was going to happen, thank you. What is going to happen is that you'll come up, you and one person from each side. I do kind of keep score, right, for the sides. But it's not quite fully Jeopardy, because this is not one, it's a multiple choice. Okay, so there's the statement, you don't have to pull it all out of your head. Thank goodness, I know I just, yeah. You guys, even while taking quizzes, somebody in this room, several people in this room are very vocal. All throughout the quiz, I'm like, I don't know how the first test is gonna go. But in any case, though, this is multiple choice, and we'll have one person from each side come up. The first time around when we go through, you're gonna wanna write your name, don't sign, just write it. I need to see it, be able to read it. And we just do that first time around. But we'll go through all of these, right? So you might be up several times before we get through all of the questions. I've got my who's who, that's more of my first unit, right? So the first module, the chemistries, which is the second module, both of these, prokaryotic cells versus eukaryotic cells, as topics, which is the third module, and the all acellular, which would be the fourth module, okay? Won't lie, it doesn't, I tried to make it a little more harder as you move down in points, but, reality is that, I just have to come up with questions. So, some questions you may go in, but that's a 1 pointer, why, that's really hard. And I was like, I'm just looking for questions, okay? When you do come up, I'll read out the question. You guys will pick what you want to do. Who you're competing with will pick what you want to do, and then it's the first one who answers. Okay? So I don't have any buzzers or anything like that. There are times if I'm looking down, I may go, who answered? If you answer at the same time, we'll just default it as no points anybody. Okay, because what difference does it make? It doesn't, going up the same amount on both sides isn't going to help anybody. If you get it wrong, you guess, right? And you get it wrong, that's fine. I will give the other person a shot to give their guess. Okay, because again, it's not true Jeopardy, and honestly, you don't get it wrong, we don't care. We're not judging each other. This is just an opportunity to get an idea of what type of material, what type of information you should be able to pull from this material, and just kind of get you going with your study, okay? All right, and quite frankly, I didn't think many of them were that hard, actually, so, maybe my test might be harder, I don't know. I don't know, when I'm sitting down making questions, and I'm going, I like this question, I like that question. Sometimes, I go, these ones, some of these might be very basic, as far as I'm concerned, but hopefully we'll do well. So, we're gonna start at the front, yeah? And come up, so two people, you, yeah. Just stand up. Go ahead and sit right up here. It's okay. This is what I do for my activity. All right, again. No stress, no worries. There's no timer. A buzzer would be fun. It's supposed to just be fun. Well, what would you guys like to be tested on?
We want an easy one.
Which topic? Chemistry, okay. And you go with what number? 100. All right. It's a picture. The atomic number is equal to the number of
B.
And then, I think I heard B over here first.
Yes, you did.
All right.
Keep it
Next.
going.
Next on the list. Yep. It should have grayed out one. Of course. All right. I'm gonna have to, no, I can't because of the fact that, let's see. 1, 2, 3, 4, 5. Give me a second here. And then,
You want me to do it?
it didn't do something that it should have, so I'll have to revise that slightly, because I should have crossed out that, so we didn't reselect the same one. Okay, what do you guys want to do?
Chemistry again.
4?
300.
Yes, I like people who take a little chance. This level of protein structure is not
Oh, D?
susceptible to protein denaturation. You gave an answer,
Yeah.
and you gave what answer?
I said D.
Okay. That was wrong, so what was your answer?
Oh my god. So B?
Nope, it is A.
Oh.
What's A? Why? That's a good question.
Because that's the order of proteins.
So, the primaries, this, I talk about protein denaturation. The primary is the peptide bond linking the amino acids. Okay, that's a covalent bond that is very strong, and just heat or acid base is not going to break that bond. These ones, all of these, in them they have ionic bonds, hydrogen bonds, all of which will be affected by. Okay? And yes, tertiary does have a covalent bond. What is the covalent bond of tertiary structure? There is a covalent bond there, what is that bond called? Disulfide bridges. Disulfide bridges, right. That is in the tertiary level, and it is a covalent bond. So it doesn't break very easily, no. But there's also hydrogen bonds and ionic bonds in that level too that will. Okay, so denaturation is still there. Okay.
Isn't C the most susceptible to go to denaturation?
No, not the most susceptible, no. C, all of them, D, C, and B are very susceptible to denaturation. Yeah. I don't have the answer to that. Okay, so here's the next one. And what do you guys want to do?
I wanna do
Who's who, okay?
What about you?
That's fine.
So which who's who are you gonna do?
What do you wanna do? Money wise. Let's go. Let's do
300?
300 also. His experiments were the first to demonstrate more complex organisms originate from other organisms supporting biogenesis.
B.
You're going for B. That is
You know this
incorrect.
one.
I think it's A.
It is A. It is A. Yep.
The worm. No worms.
They all of them were there.
What are you feeling? I want to go risky. Let's go 500 or something. Okay.
Eukaryotic 500. That's gonna be an easy question. Everyone else is gonna go with 500. Cell wall composition of fungi cells.
E. C. Oh, sorry.
I heard E first, E is correct.
Okay.
Who's got these? Who's got this? Plants? Bacteria? Archaea, right? Is the pseudopeptidoglycan. I heard it, I heard somebody say it. Algae, yeah, the algae, algae, and then fungus.
Agar, it's the
Agar
one used for
agar
the laboratory, right?
Which gross is. All right. And that was.
Honestly, I have no clue what I wanted to do.
I don't care, honestly.
I guess it's 200 or 2.
What's 200? Did I not write down, was that
Sure.
200 was the,
No, we did a 300, you did a 500, and then after you did a 100.
the 300 was the, you're right, I didn't have, we were talking and I didn't have that.
So why are we not?
Okay.
Sorry, am I back here? 400.
His work demonstrated the existence of N to spore for some bacterial cells.
D.
And you said D?
Yeah.
That is incorrect.
Maybe E?
E is correct.
God.
This question gonna come on? I'm sorry.
Didn't want to say the same question.
Eukaryotic? Prokaryotic? That's fine.
Oh.
300. All right. This waxy molecule is incorporated into the cell wall of acid-fast bacteria.
D?
Is it?
D is incorrect. A is incorrect.
Okay.
B.
It is actually
C?
That's glycolic acid. And we're actually gonna be, looking at that again today in
Instead of just copying all that.
Okay, so I've got 1 and 2, and you wanna come up so you can sign. Once everyone's signed, you can stand up where you're at is fine, so that I know who's playing, right? But you don't have to actually come up if you don't want to. Just need a coming up for this.
Sorry, it's a crowder.
All right, what do you guys think? Kind of have to, unfortunately, I'll fix it so that next time, but yeah, or hopefully I'll be able to fix it. I know I had it one time.
Who's 2 for 400? Who's who?
Who's who for 400 has already been done.
Oh.
So we have 500, who's who, you have 1, 2, or 5? You wanna do who's who?
Five?
5, okay, 500. You're not risking it all.
Come on.
He discovered all this antibiotic given the name penicillin.
Oh, I know that.
Oh, no. Wait,
You know that we.
I know this. I think it's D, yeah.
You're saying D.
I'm saying
And
D.
you are correct.
Oh, I had to think about that one. I had to go each one, I'm like, no, yeah.
Now, did you guys switch? Okay, you did switch. Shoot, so do I.
You wanna get them all right?
Yep.
Which topic are you looking at?
Prokaryotic.
Prokaryotic, you're right, you got 1, 2, 4, and 5.
Let's do 200.
200, okay. I'll go with 200. This bristle-like structure can be found on some bacterial cells and serves for attachment to surfaces.
Oh.
Not C.
B?
It's not C.
Oh, B?
It is Not B.
Oh.
It's A. It's Well, yes, it's a little easier when you're not on the spot. All right, so you're up, and then you can just stand up. She has to actually, write her name on this. Right, and at this point, and everyone's written their name, right? Yeah. Okay. All right, so, which topic? Which topic?
Nobody's done. Yes. Acellular for 100.
For 100.
Does that work for you? Okay.
Just don't answer at the same time, okay?
We'll work on that.
Viral structure which is not present in all viruses.
C.
C is correct.
Oh, sorry. I wish it was more, guys.
So, you're up, yeah. You can stand where you're at, it's fine. So, just stand though, so I know who I'm trying to make sure and what are you guys thinking? Which topic? Who's who for 100? All right. Healing to bacteria to disease anthrax. C. You said D is correct.
Oh, it is?
Not the collar, I'm guessing. All right, just stand where you're at, it's fine. And what topic you guys want? You want to finish off the who's who? There's only 200 left. Okay, we'll finish off the who's who. Okay. He never found the magic bullet to kill all microorganisms.
D?
Not D. What's your? And you're going with B? No, it is C.
Yes.
It's all air.
Rich and rich.
All right, so that puts us to chemistry, prokaryotic cells, eukaryotic cells, and aviral are topics. What you guys got on topic-wise?
You can pick whatever you want.
Oh, shit. Okay, sorry. Prokaryotic 400.
That's exactly what I'm thinking.
Cool.
Prokaryotic for 100. Let's see what I got here. This crystalline structure stores nutrients for the cell. Not A.
I think it's D.
D is correct. Okay. Which topic?
Chemistry 500.
Chemistry 500. And she's over there going, take back, come on. This type of molecule increases the concentration of hydrogen ions in a solution.
C.
Yes, C is correct. That's it. That's exactly. Oh, wait, no, that was two. All right. Did you learn from her mistake or?
Let's do chemistry for 400.
Chemistry for 400.
Okay.
Apparently you didn't. No, this monomer has a pentose sugar, nitrogenous base, and a phosphate group.
B.
B is correct. He was just as you throw his beheading there. you're stand up and, okay. So what are you two going for? Do I close out chemistry for 200? Sound good?
Okay.
The power of not choice.
This functional group, two classifications based, I must have put up that, has two classifications based on where the functional group is placed on the carbon skeleton.
C.
You said C is correct. No, I'm sorry, wait, you said C, I'm sorry, C is not correct.
Oh, sorry.
I'm looking at the wrong one as I'm
Oh,
saying this. I'm sorry, so your guess is? Here's the beauty, you can pick anything and it doesn't matter, right? I mean, no, it is D.
Oh, no.
It's D. Carbonyl. Yeah. All right, so who are my next two? You're up and. All right, so we've got, in prokaryotic, we have 2 left, 4 and 500. In eukaryotic, we have 1 through 4. And in acellular, we have 2 through 5. Pick one. Prokaryotic, 4, okay? All right, term given to a tough or group of flagella on one polar side of the bacterial cell.
C?
And it's not C.
We both did C, I think.
Yeah.
Oh, you both said C at the same, I only heard it once, so you must say at the same time. So it is D. Amphi, here's the thing is, amphi, when you think of amphitheaters, amphi refers to 2 sides. This is coming from one side. So amphi is fine for the tough aspect. It could be 1 or more, but then it's coming from both sides, okay? Alophotriphalus is where it's coming from only the one side, and it's a group of them. Obviously, this is only one, and so it's coming from one side. And paratriculus is where it's all throughout, all over the surface.
So you said it was D? It's D?
The answer is D, correct. And clearly I have no, this is not the one that, the other one is the one, this one. All right. So, who am I at? So you were we're back in front for you guys. Yep, choose the last, and so we're here, yeah, there we go. And we have, you close out prokaryotic cells, where we got several in both the eukaryotic and the virus.
She's like, no.
Eukaryotic, 100.
Eukaryotic 100, all right. Let's see what I got. This organelle stores the genetic information.
Nucleus?
Yes. Which is
C.
C. Yeah. All right. Prokaryotic 500. All right, we're gonna close out that one. Okay. And our question is, this structure functions as an endotoxin, causing symptoms as fever, hemorrhaging, and septic shock. And the type of cell with this structure are so it's the molecule and the type of cell. The molecule that is a endotoxin causing, symptoms for the patient, and which type of cells have that particular molecule. You're saying D? Yes, D is correct. Lipopolysaccharides, and they're found on gram-negative cells. Okay, so that puts us at, no, all right. Now who's my next? All right. So we're down to eukaryotic or cell or viruses. So we acellular 200. Mark it off now. Yeah. And viruses are only organized into
D.
D is correct. You did say, you're the one who said D, right? Okay, just want to make sure. As soon as I look down, I'm going by sound. Okay. Just families. Now, we have 2, 3, or 4 in the eukaryotic cells, and 3, 4, and 5 in viruses. Okay, slash acellular, actually. Go ahead. 300. This step or steps of viral replication in an animal host does not occur in viral replication in a bacterial host cell. Steps occur in these steps occur in the animal virus, but not for the bacterial virus.
E.
Say it again? E. E is correct. Right. Uncoding and budding. I won't lie, D and B sometimes sound very similar when people are saying that, at least from my front area, which is why I've asked that for clarification. All right. What do you want?
Go ahead, Biscuit.
I'm over here like gamble.
Acellular 500.
What is not true about the lysogenic cycle? What is not true about the lysogenic cycle? E. You're saying E? That is not correct. D. It is C. It is C is the correct answer. It is not replicating, it is not producing more viron during the time that it is part of that, as a prophage. All right, so, yeah, we've got, all right.
You can go ahead. Go
What do you
ahead.
want?
It doesn't matter.
Eukaryotic for 400. All right. Location where ribosomal RNA biosynthesis occurs and the ribosomes are assembled.
C.
C is correct. Nucleolus. That's what.
Make us proud.
Thanks for these points. You don't have to write Please.
So I've got 2 and 300 for eukaryotic cells and 400 for the acellular. Which one do you want to do?
Acellular 400.
Acellular for 400. All right. This is not a way that viruses can cause human cancer. You're saying B, no, that is a possible way, so what is your guess? I already know it's a guess. You're saying E, no, it would be D. D for the abnormalities in the cytoplasmic membrane is not the cause. All right. And then we have two more left. There are eukaryotic cells, number 200 and the 300. What do you guys want? 300, 200?
3.
All right. Eukaryotic, 300. Membrane where the green photosynthetic pigment chlorophyll is
C.
found. You said C?
See?
Good job. And then, so we're down to our last one. Who's my last? 300. I think you're up.
Me?
Yep.
Oh, goodness.
You're gonna say, I've never seen you in front again. All right, so, what did I say? There's 200 before the eukaryotic. This structure is not part of the endomembrane system. This structure is not part of the endomembrane system.
B.
B is correct. All right, that was it, guys. Your torture is over. You're gonna end lecture early, that's fine, we'll start lab early. We have two labs, but three different stains we're gonna be doing. So leave it and get in there 15